I'll give everyone a moment to look over our session 2 outline for ingredient considerations. Here's what we have coming up in the next hour and 15 minutes or so. I will be breaking it up with some questions here and there, some polling questions. But first up for session 2, we have new ingredient pain points with Ray. Ray Matulka is on the line from Burdock Group. Ray, are you with us?
Yes, I am, Sarah. Thank you.
All right.
So, I am with Director of Toxicology at Burdock Group. It is a consulting company that is focused on assisting food ingredient manufacturing companies in assembling documentation to show that a new ingredient is safe under the intended conditions of use. So I've been with Burdock Group for about 15 years, and Burdock Group has been working within the ingredient manufacturing scheme for almost 30 years. So we have seen quite a bit of information that has gone through and some issues that can come up concerning feed ingredients. So I just wanted to talk to you all about some of these issues that can be encountered and what you can do in order to show safety of your new feed ingredient. Next slide, please. So what I will be talking about is about 4 different points. First, I want to talk about variations in approval processes. Why would a company be pulling together such documentation, and who would they be presenting that documentation to? And then getting more into some of the points that you need to look at and think about concerning a new feed ingredient, especially for pet food, in characterizing the intended use of the ingredient. That was touched on a little bit by Carrie and Brett earlier. And then to talk about the characterization of the new ingredient, not just the intended use. And finally, then talk about a little bit more on international approvals, as we are a international type of industry. So sometimes international approvals outside of the U.S. can actually affect the intended use in the U.S. Next slide, please. So first off, I wanted to talk about the different processes that are necessary to go through in order to get a new feed ingredient, pet food ingredient, approved, or at least appropriate for use in the animal's feed. Now keep in mind, I want to first talk about the federal level, because federally, the Food and Drug Administration reviews products that cross the state lines, interstate commerce, whereas the states mostly deal with what occurs inside their own state lines, or intrastate commerce. So at the federal regulations concerning all 50 states, those regulations state that any ingredient that is reasonably expected to become a component of food is a food additive subject to pre-market approval. So this means that if you're going through a food additive petition such that you have to submit your information to the FDA, it is considered a pre-market approval. Now this occurs if the ingredient has not previously been determined to be generally recognized as safe or meeting this so-called GRAS status. Now, companies in and of themselves can reach a GRAS status and document that their ingredient is approved or appropriate for use in pet food under the intended conditions of use. And they can go through and notify the FDA in order for the FDA to understand that that product is out there and is available for use. That's not mandatory, but it is helpful. Now, the states, as Carrie and Brett also talked about, also have the AFCO process. And within this process, when you submit a new definition for a new feed ingredient for pet food, the data that is necessary to show safety is actually submitted to FDA in order for the FDA to review it and agree upon the safety. So as Carrie and Brett had stated, there's a lot of interaction between the states, the individual states, as well as the federal government to make sure it is a more or less seamless process to be able to get your product to market. Now, I also have down here safe and suitable, in that the USDA also looks at meat and egg products, among others, and some of those meat-containing products for pet food may be coming from actual human food-based facilities, as was being stated earlier by Gary. And the USDA actually looks at a lot of technical ingredients that may end up in pet food to determine if they are safe and suitable. So these are all different ways and different parts of the regulatory process that a new ingredient manufacturer has to be cognizant of as they're going through and developing their new ingredient. Next slide, please. Okay, so I now want to move from more of a general overview of regulatory aspects and who you need to send the documentation to and who reviews documentation, more to what types of documentation are necessary, how you need to look at your new ingredient, and see what is available and how it is to be utilized. So that's first the intended use of the new ingredient. Again, going back to Carrie and Brent, when they were talking about labeling, what types of label claims you can utilize. There are several different types, but for pet food, most often you're looking at new ingredients that are going to be added as a nutritional part of the diet for dogs or cats, other pets. And so in that aspect, you have to understand and promote your ingredient as a nutritional product, providing fat, protein, caloric content such as that, or in terms of a defined nutrient, such as vitamin D, vitamin E, other types of nutrients that are defined. Now, other types of ingredients are added to pet food for its technical effect. So the technical effect can actually be as a stabilizer in the feed, or as a thickener, something like that, in the fact that the ingredient has actually a technical effect on the pet food. In terms of intended use, the ingredients are approved for use within both the federal and state regulatory processes. The ingredient in and of itself is not approved, but it's actually the intended use. And the intended use includes the anticipated amount that's going to be consumed by the animal. So you have to really understand how much is going to be added and explain that to the regulatory bodies in stating that this ingredient, let's say it's a vitamin D, is going to be added to the diet at 50 ppm. Now, keep in mind, there are some ingredients that are nutritive at low levels, but actually can be toxic at high levels. And that's really the reason why this intended use is here. For example, copper is a known metal that is needed and is an essential nutrient at very low levels. It's used in a variety of enzymes, as well as in the blood in the heme. However, at very high levels in all sorts of animals, including dogs and cats, high levels of copper can produce toxicity. Therefore, you really have to understand how much is being added and what is the necessary levels needed by your dog or cat to be consumed. The other aspect is the structure or function of the body. FDA has stated that the intended use indicates what can be stated on the label, and FDA has historically stated that ingredients that affect the structure or function of the animal body are drugs if they are not nutritive in nature. So you really have to be careful on how you evaluate what is going on and what is the intended use of your ingredient and how it affects the animal's body, because you really want to focus on the nutritive aspects of any new ingredient. Next slide, please. Next off, we want to characterize the new ingredient. Many new ingredients in this day and age are produced through a variety of ways with a number of different raw sourced ingredients. If you, let's say it is a new ingredient that's taking the previous example of a vitamin D analog that is produced from a fermentative process, and it actually is absorbed into the body into the pet's body at higher levels than typical vitamin D that's found already in the industry. In order to provide adequate information concerning the safety of your new ingredient, because this is a variation on the manufacturing process, as well as the biological activity of this ingredient, you really need to submit documentation to either the FDA or to AFCO, or either provide a generally recognized as safe or GRAS documentation showing that you really have evaluated the safety of the ingredient and have concluded that the ingredient is safe. The source of the ingredient is really key on many of these fermentative processes, because you have to show that this source ingredient, if it's a fermentative microbe, that microbe actually does not produce any harmful, toxigenic, or potentially pathogenic aspects to that source microbe. And if the microbe has been genetically modified in order to produce this vitamin D analog, then you really have to explain to the FDA and other regulatory bodies that these genetic modifications do not adversely impact the overall safety of the final ingredient, the vitamin D. Now, moving on from there, you really have to explain also the manufacturing process. Manufacturing process, really a lot of that had been gone over by Gary Smith earlier. It is part of the FSMA, and in this process, a way of explaining how this ingredient is manufactured is similar and analogous to the different parts of FSMA, such that you have to explain the safe use of technical ingredients. If you use any solvents, that those solvents in cleaning up and purifying the ingredient are safe for use in animal feed, because every ingredient that goes into the production of a pet food ingredient has to have been approved. And that does include any, as I said earlier, texturizers, binders, solvents, humectants, preservatives. All of these ingredients really have to be considered and approved to be safe in one fashion or the other to be utilized in animal feed. You also have to have a complete understanding and analysis of the whole ingredient, not just the active form. I get presented with many technical sheets that indicate the active ingredient, let's say in this case vitamin D analog, at 10% of the overall product, however, of this ingredient. However, all of the rest of the 80% or 90% of the formulation is stated as being inactive. This is just not quite sufficient for the FDA to understand what is going on and what is included in this ingredient. You really have to analyze the whole ingredient formulation. Let's say for this microbial sourced ingredient, it may have additional amounts of protein, it may have carbohydrates. You have to analyze those different pieces and explain what it is. All of this needs to go into a specifications sheet, basically a table explaining how you define this ingredient formulation and what goes into it, what are the specifications? For, in the case that I'm carrying on, the example, this would be the vitamin D analog. You have a validated methodology in order to analyze for vitamin D in the ingredient, and also you're able to analyze for any um, protein, carbohydrate content, things of that nature, ash, in order to show approximately 100% of what is in your whole new ingredient formulation. These specifications really have to be reproducible and define the ingredient. FDA regulations and actually guidance documents do indicate that you need to also show consistency in being able to produce this ingredient. As such, you have to show consistency, which means making a number of different batches of your new ingredient to show that you are consistent in producing the vitamin D analog as you have stated on the label. In addition, the statement on the label has to be accurate and truthful throughout the stability of the ingredient. Not only packaging of that ingredient when it's from a business-to-business type of sales point, but also when that ingredient is utilized in the pet food itself. So many businesses, as you're preparing your ingredient for sale or preparing your ingredient to be added to the pet food, you will have to characterize this ingredient in terms of how stable is it for the lifetime of the ingredient formulation as it's sitting on the shelf, as well as the lifetime of the ingredient when it's added to pet food. Because the FDA and other regulatory bodies, as third-party type of evaluations, which they do conduct from time to time, they will pull pet foods from the shelf and analyze for different components of the ingredients list to show and to confirm the minimums and the maximums that Brett had earlier talked about. So this is a very critical phase on showing that your new ingredient is safe and is appropriate for its intended use, as well as time frame of use. Next slide, please. Now, many times going through and working with a lot of different companies, many companies are international based and would like to bring their new ingredients or their ingredients that are already approved within, let's say, the EU or Australia, Brazil. They would like to bring those ingredients to the United States. Such a large pet food industry that we've got and large number of consumers, I understand. However, a lot of these companies and any companies that are producing new feed ingredients have to understand that getting approval outside of the United States in a different regulatory body does not automatically approve the use of that ingredient within the United States or within state agencies. There are a variety of reasons for this assessment. Many times the safety or the utility data may not be publicly available that the, let's say in this example, the EU has looked at, such that the US cannot use it for a generally recognized as safe assessment because the critical data was not made publicly available to the extent that scientific regulators or the scientific community is able to analyze and conclude that it's safe. In other times or other experiences, the European Union actually looks upon herb-based products, botanicals, as being of a lower safety concern than other synthetic evaluation or other types of synthetic products. And if that's the case, many times the European Union has made inferences that because it is a botanically based product, the quality and the quantity of data may not be as rigorously required as the US, the FDA would require. And so in bringing this ingredient over to the US, you really have to understand what types of data the US would require in order to prove safety in a pet food ingredient. Pet food ingredients are also approved for safety within the US for pets. And so ingredients that were utilized in the EU for commercial livestock, for let's say cattle feed, may not have the amount of data or the quality of data to show that it is also safe for use in, let's say, cat food. There are variations within the physiology between livestock, cattle, swine, versus some pets, such as cats, such that there is not a direct read across between the safety information from one animal in order to show the safety and use in another animal. Third point that I wanted to point out concerning international approval affecting the intended use in the US is really concerning marketing statements. Now, Carrie and Brett had talked about previously the use of labels and label claims. Keep in mind that in the EU, Australia, other regulatory bodies, those regulatory bodies may allow certain claims on labels and in brochures, marketing brochures, that actually would be indicative of the ingredient being utilized for drug aspects or claimed to be a drug here within the United States. So really you have to be careful in terms of websites and how they are directed. If a European website is directed towards the US, you really should have a US focused website such that the marketing claims are not directed towards things that may be considered a drug-like effect here in the States. And also marketing brochures need to be very clear that the intended use of the ingredient fits within the US regulatory pattern. Next slide, please. Now, I do want to talk a little bit about imports, exports of feed ingredients, because really this does also hinge on approval of those ingredients, and the components that are used to manufacture those ingredients must be approved within the United States. This is one thing that international companies can find very difficult, just in the fact that any ingredients being shipped into the United States, any formulations, must also meet approval for use in the United States. This includes any technical additives that may be used in the production. As such, the Food, Drug, and Cosmetic Act does state that foods and feeds must be safe and wholesome. They must contain no deleterious, harmful, or unapproved substances and be truthfully labeled. It is that unapproved substances aspect that can be difficult. As I said, even something such as a solvent utilized to purify and condense the ingredient formulation must be approved for use inside the United States. Now, I would go through the foreign supplier verification program, but Gary went through that very clearly and cleanly. It's just the fact that really there has to be some verification process that the ingredients being produced do meet U.S. safety standards, and there does need to be a prior notice of any imported food, including animal feed. And finally, if your ingredient is being derived from plant or animal imported materials, you actually may need to speak with the USDA to obtain a permit for that aspect. Next slide, please. And so finally, I just wanted to summarize, in fact, that you want to really focus that your ingredient, as you're bringing it to market, as you're actually producing it and going through the initial stages, you have to fully characterize the ingredient with any notice of limits on contaminants and stability of the functional ingredient when added to the feed. You have to show the ingredient to be safe and meeting all regulatory requirements. And you have to make sure that your intended use would not be evaluated or thought of by FDA as to having a or considered a drug-like effect. Finally, ingredients imported must still also contain only ingredients that are approved for use within the U.S. Thank you very much.
Awesome. Thank you, Ray. All right. So next up, we have one of our most requested topics, contaminants. So we have John Ruther on the line from our Eurofins Central Analytical Labs to discuss various contaminant risks.
Thank you, Sarah.
John.
And I'm the president of Eurofins Central Analytical down here in New Orleans. Our laboratory is responsible for identifying and testing various chemical contaminants in foods and feed products in commerce. And particularly the ones that we show here are considerations for risk in pet food products and ingredients. Mycotoxins, histamine, drug residues, heavy metals, pesticide residues, dioxins, and PCBs. Next slide. We won't cover all of the individual risks that we just mentioned, but some of the more significant risks and more common risks that we see in ingredients are going to be talked about and discussed today. Mycotoxins are a risk that we see in many of the ingredients in the food industry and pet food industry. And some of the ingredients that we commonly see mycotoxin risk here, especially high mycotoxin risk, include peanut meal, which has been shown to have, or at least in historical terms, have had high risks for aflatoxins, and those are probably a lot less risk than they have been in decades past. Corn-based products show a lot of risks for aflatoxin, DON, and fumonisins. Occasionally, there'll be zearalenone risk. It's very, very crop year dependent. It depends on weather conditions and how those products are produced. Whether there's severe drought conditions, there can be elevated concerns for mycotoxins. The more common ingredients in animal feed and pet food would be ingredients like cornmeal, dry distiller grains, corn gluten meal, corn gluten feed pellets. Of those ingredients, dry distiller grains are probably the most highest risk because the corn product itself there is relatively concentrated over the originating material, which is whole corn. In some cases, dry distiller grains can be concentrated by a factor. Mycotoxins can be concentrated by a factor up to 3 to 4 in that product because of the removal of starch from the product. And many of the mycotoxins, particularly aflatoxin and DON, will stay with the eventual end product, the dry distiller grain. Sorghum, which is not a more common ingredient in pet food, is a high risk for zearalenone. Next slide. Some of the ingredients that have lower mycotoxin risks, and mainly because they do not occur at very high concentrations or their occurrence of this in particular ingredient in the final pet product is low, would include the following: wheat, which has normal levels of DON and ochratoxins present. Several of the botanical products have aflatoxin and ochratoxin present. Spices, which are used sometimes in pet food products, may contain aflatoxin or ochratoxin. Corn oil and peanut oil do have some small risk for aflatoxin and zearalenone. The rice, barley, and oat cereal complexes will sometimes have don and ochre. And then soybeans have a very, very small risk for ochratoxin. Next slide. So published limits for mycotoxins and other contaminants are sometimes sparse in the U.S. In U.S. commerce or U.S. trade, but U.S. Food and Drug has set limits for pet food for these three mycotoxins. Aflatoxin is 20 parts per billion, DON is 5 parts per million, and fumonisin is 10 parts per million. In normal healthy grain supply, including corn, these limits are relatively easy to meet. Sometimes in difficult years, they are less easy to meet corn, particularly in DDG products. Next slide. So I'd like to talk a little bit about considerations of methods used to determine these mycotoxins. There are many, many method platforms available out there, both for screening and fast, quick analysis in the field, as well as reference methods that can be used for finished products as well as ingredients. ELISA-based kits are very prevalent in field use and in laboratories as well, in field laboratories. They have several advantages. Probably the biggest advantage is that they are fast and easy to use. We have seen them used in analysis of truck samples, although they probably could be a little faster for assaying truck products on the spot. But you usually can get a result within 40 to 45 minutes using an ELISA kit. If you're doing just one test at a time. They're relatively cheap to execute. You only need a reader to quantify mycotoxins such as aflatoxin or DON or fumonisin. And they're relatively accurate. The precision is okay. It's not the greatest, but it is acceptable for most screening testing, especially in the field. USDA has approved many mycotoxin kits based on ELISA technology. The disadvantages is that detection limits are sometimes compromised depending on the ingredient, but detection limits are sometimes not as good as what you would find in a reference method. And if you are under a contractual specification with a producer for mycotoxins, sometimes the detection limit is near the contractual specification, which causes trouble especially with respect to the variability of the kits. The level of precision near the maximum limit for some export countries is also a problem. As I mentioned, while the precision is acceptable for most normal levels of toxins, when those levels occur near the maximum limit, the precision of the kit may cause issues when trying to resolve whether or not you have a pass or fail ingredient lot. Accuracy of the kits is very dependent on kit shelf life, and that kit shelf life needs to be monitored with controls. Keeping a large quantity of kits in stocks can be expensive. The shelf life of kits is limited. Next slide. So these are some typical values that we normally see in various products, particularly corn. The test range of ELISA kits usually meets those typical values and also meets the requirements of the food and drug limits. Next slide. LCMS methods and HPLC methods in general are gaining more popularity in laboratories. The advantages of these methods is that the sensitivity of the instrumentation is usually much higher than ELISA and usually exceeds the government recommended levels for safe feed ingredients. Another very big advantage is that some UPLC methods can run a full toxin suite in 10 to 15 minutes, which tremendously increases the cost efficiency of running these tests and the throughput. And the biggest advantage probably is the fact that the interferences that are sometimes found with ELISA or other field-type methods is significantly reduced with HPLC or LCMS methods. Trends in the industry is typically internal standards are used for controlling recovery in HPLC and LCMS methods, and now internal standards are available for all the common mycotoxins for LCMS analysis. And then methods that are evolving towards multi-toxin analysis. We've seen methods available that can analyze for up to 25 different mycotoxins that are commonly found in cereal grains, and these can be done in a single analysis. Although we have found that some of the more complicated ingredients like corn gluten feed pellets cannot be analyzed successfully with multi-toxin tests. Next slide. The targeted mycotoxin methods that we use for HPLC are typically aflatoxin and ochratoxin using Amino Affinity columns with LCMS or HPLC. And the MQLs and the precision levels are quite good with these. MQLs range from 0.2 to 8 ppb depending on the matrix. Next slide. This is an example of an LCMS trace, and it shows you the level of selectivity of the test. Each individual component of the aflatoxin is measured separately, so that's an advantage over ELISA techniques. So you can see the B1 clearly in this trace. And also the bicolored nature of these chromatographic piece shows you that not only a quantitative signal is obtained with this method, but also a confirmatory signal is obtained. So it's very clear that what you are measuring is a mycotoxin and not some false positive. Next slide. Another targeted mycotoxin method that we use very commonly is fumonisin, and like aflatoxin, we use Amino Affinity columns for that. Again, recoveries are quite good at the levels of concern. This particular chromatogram shows fumonisin B1 and B2, but we're now currently able to also measure fumonisin B3, which is the third component. And although it occurs at much lower levels than B1, it's still an important contributor to total fumonisin. Next slide. Another mycotoxin method that's quite productive is the Romer MultiStep 226 column for vomitoxin, zearalenone, T2DAS, and HT2. We can determine all of these components. As you see in the chromatogram below, they can all be separated very, very well and isolated with both quantitative and confirmatory peaks. Recoveries are quite good. It's very sensitive, typically 1 to 10 ppb for individual mycotoxins in this range. Next slide. The next important category of risk in ingredients is histamine. These would be commonly found in fish products. The most highest level of histamines occur in fish products which have high histidine levels, which is the precursor for histamine formation. And those would occur in these types of open water fish, such as mackerel, amberjack, tuna, sorry, sailfish, marlin, which have very high histidine levels, as you see. These are not histamine levels. These are histidine levels that are common in these fish. And then also at a lower level, anchovy, sardine, mullet, menhaden, and herring. So ingredients coming from these types of fish products should be very much a concern and consideration for histamine testing. Next slide. Lower histamine risk would be more of the freshwater fish and some of the estuary type fish, salmon, trout, I'm sorry for the spelling there, but it's trout, sole, and cod, which have histidine levels of less than 2,000 parts per million. Next slide. Histidine action limits should should be taken into consideration when testing. Testing should always be performed on incoming lots of product. The analytical testing should complement sensory testing and temperature control of the product. These are FDA requirements. Also under consideration, the high variability of histamine levels throughout the lots and within the various sections of individual fish need to be considered when developing a testing plan and a sampling plan. The FDA limit of 50 parts per million is based on a composite of 18 subsamples per lot. So within those subsamples, there can be variations and large variations, but the composite needs to show less than 50 parts per million for it to be accepted within Food and Drugs limits. Next slide. Some of the more prominent histamine testing methods are in place now. There are two reference methods, AOAC 977.13 and ISO 19343, promulgated 2017, which is an HPLC method. The AOAC method is fluorometric based, and it's in common use in the USA in many laboratories. Then finally, a field method, ELISA methods that are produced by R Biopharm and Neogen are very suitable for field use. Although the same types of concerns that we saw with mycotoxins in terms of precision are still prevalent in ELISA methods. Next slide. Moving on to another risk area is drug residues, and I won't spend a lot of time on this, but there are quite a few classes of drug residues in the markets these days in ingredients, particularly meat ingredients, but the highest levels can be found in the organ products of pork and chicken, particularly tetracyclines and sulfonamides. In aquacultured fish, there are drugs that are banned in the U.S., banned from use in the U.S., and any residues of those drugs are considered violative of U.S. Food law. Fish meal produced from these aquacultured fish could be another source of high-risk drug residues, and fermented products from outside the U.S. Antibiotics are commonly used in the fermentation process to control bacteria, and those antibiotics can be found as residues in the product that you are using as an ingredient. Next slide. The lower drug residue risks occur because these particular residues either occur at low levels or because they're not organs. So general meat supply is generally well controlled in the U.S. Meat supply. Meat from euthanized animals may contain illegal drugs, but because of the low risk of occurrence in the pet food, we consider this to be a relatively low risk in the food product, in the final food product. Poultry meals, relatively low risk, low levels of drugs are found typically in poultry meals. In U.S. aquacultured fish, the same applies. Where drugs do occur for these approved drugs, they occur at relatively low levels. And of course, dairy has risk for drugs, but low risk. But you also have to consider imported dairy products may have a risk from melamine contamination. Next slide. So the USDA actually assesses or asserts limits on U.S. meat and food products for approved drugs in use in the USA. FDA, on the other hand, provides limits on imported aquacultured fish products, and these limits include zero tolerance on those banned antibiotics that we just mentioned, and those would include chloramphenicol, nitrofurans, fluoroquinolones, and quinolones. In recent years, these have been brought into better control from our foreign supplier partners, but they are still considered a risk. Next slide. Analytical methods for drug residues include the same types of methodologies we've already discussed. LCMS is used very commonly in reference laboratories to test class-specific drugs. And also FDA, USDA multi-residue methods can be used for broad screening of multi-class drug analysis. ELISA or CHARM methods tend to be more single residue specific or class specific, but they're very, very cost effective. And if you have a specific targeted drug in mind that you want to monitor, that can be very, very cost effective. Next slide. So in summary, when considering methods for analysis of chemical contaminants, there can be a high degree of selectivity with respect to high and low risk ingredients. There are many options available for cost-effective monitoring of contaminants once you make your risk assessment. And in general, reference laboratory methods that we discussed today are optimized for analytical performance rather than cost. But the end result of that is that they're quite reliable at low concentrations. And these methods can be modified for a specific ingredient to optimize their analytical performance on that ingredient, whereas the ELISA methods and the other methods are relatively less able to be reliably modified to optimize them for an ingredient. Thank you for your attention today.
Thank you, John. Really quickly, before we move into the second half of our second section, I want to launch a polling question really quickly for our audience, just to gauge about halfway through our digital conference today. How are you feeling about our different pain points? Which are of the greatest concern for your facility? I'll give everybody about 1 minute to respond. Our last poll only had 50% participation, so let's see if we can beat that today. All right, I'll give everyone just a few more seconds. We're right at the 50% mark. I really feel like we can do better than that. Okay, I'm going to close our poll and I will share the results with everyone. If you did submit other as your response, shoot me a message in the question box or the chat box and let us know what it is you're worried about. We do have other topics in our session. I just couldn't fit them all into our options here. But it looks like most people are worried about meeting just the general FSMA requirements. So keep submitting those questions. I hope that Gary gave you a good overview. And I'm not sure if we mentioned before, but questions that we don't get to answer in our live session today, we will follow up directly via email with you. So any question you ask today, you are going to get an answer. It might not be live, but it will be soon. That'll give you the opportunity to continue the discussion with some of our panelists on the line today. So with that, I'm going to take down our results, and we'll keep moving into allergen and GMO label claims with Pierce. Pierce, are you on the line?
I am here.
All right.
So let's move on to my first slide. So that's me. I'm the lab manager, or one of the lab managers here at Eurofins GeneScan, which is the molecular biology unit from North America. And our interface with the pet food industry is mostly involved with supporting voluntary marketing claims that create analytical problems for you and your customers. So what I mean by that is really trying to exclude problematic ingredients from your finished product, be they monitoring processing in-house to make sure your changeovers are clean, or that you're keeping separate lines actually separate. We're trying to exclude GMO crops, for example, and then control allergens, both traditional human allergens that Gary touched upon earlier, as well as sort of allergens in pets, which the science is less clear on. Next slide. So when we talk about GMO, traditionally we've really been talking about row crops, and the big three are corn, soy, and canola. So pretty easy for most pet food manufacturers just to exclude those entirely from their products. But you have some other items like beet and potato, alfalfa, also rice, that may be in there intentionally, and those products do exist in transgenic form. So an issue for the pet industry if you're trying to exclude either for export or for making a private label claim. And then of course we have on the horizon sort of new products that no one's really considered in the past, like the aquabalte salmon, which is engineered to grow twice as fast as the regular fish. Obviously developed for the human market, but if somebody's processing it to put on my plate, the byproducts are going to end up in pet food pretty quickly, just as part of the value capture chain. And Ray mentioned earlier, there are a special class of GMs that are generally considered processing aids. So if you use a transgenic yeast to make a fermentation product that goes into your finished product, that may have some implication on your labeling if you are exporting or doing an organic product or some other private label organization non-GM claim. Next slide. And so when we do GMO testing in finished product and in ingredients, primarily we're using PCR, or polymerase chain reaction, which is a method that detects the actual transgenic DNA left in the product. And we can use qualitative methods that detect presence or absence of the target, and also quantitative methods, which will give us a percentage of transgenic ingredient. And that's an important concept. For example, we can very accurately determine the percentage of GM corn in a corn gluten meal, but DNA methods generally are terrible at determining the fractional percentage in a complex finished good. So while I can tell you 10% of your corn gluten meal is GM, I cannot very easily or accurately tell you 10% of your product is corn gluten meal. Next slide. So the other intersection in our office is on allergen. Gary introduced the FDA's mandates to label for human allergens. So that's the big 8 that everybody is probably aware of. And those can be monitored using ELISA assays that you may be familiar with from human food for things like eggs, wheat, dairy, soy, and any of the other tree nuts and other things. But where food allergens are well defined in humans, the diversity of different pet diets and the lack of consensus on the scientific side makes defining what an allergen is in a pet species a little bit vexing, because not only are you dealing with different animals, but a very wide variety of protein sources, be they grains or meats. Next slide. So when you're trying to source a single high value meat source in a pet food, for example, a salmon or elk, if you have access to relatively unprocessed raw material, ELISA assays tend to work fairly well, but as you're all well aware, usually you're dealing more with a rendered or highly processed product by the time it goes into a pet food. So ELISA methods tends to break down just because the target proteins are denatured or hydrolyzed in some of those processes. And in those situations where ELISA is not a valuable tool, you can turn to PCR, which, if you have 3 lines running in your factory, it's pretty easy to make targeted assessments of whether you're getting crossover amongst them, or to discriminate between 2 closely related protein sources, one of which may happen to be cheaper than the other. Next slide. And so in summary, there are tools that are available to manage your risks that arise from these voluntary enhanced marketing claims. The tests that you do for GMO pet food are pretty much the same tests that we would do for the human food. And allergen, be it actual FDA allergens or allergens in the sense of my dog gets hives if he eats chicken, but not if he eats beef, can be managed using both traditional ELISA methods and also focused PCR testing. So that's my bit.
All right, thank you. So last but not least, I'm going to hand it over to Megan Stike. Many of us have seen probiotics popping up everywhere across the food chain, especially in pet food products. So Megan Stike's on the line. She is one of our probiotics and direct-fed microbial experts. Megan, are you on the line?
I am.
All right, I'll hand it over to you.
Thank you, Sarah. So this is me. I'm Megan Stike, and I am the Business Development Manager for Eurofins Biodiagnostics. I do have a focus in probiotics and microbial-based products. I am a microbiologist as well, and an avid dog lover, so you can see that's one of my 2 mutts in the background. Today I'm going to be talking about label considerations, specifically enumeration, identification, and accuracy of labels. Within each of those, I'll try to dive in and answer some of these questions that I often get, understanding what is a viable count, what is the production count, how to test for shelf life as it relates to microbial-based products, identification and the specificity of identification, whether it's genus, species, or strain, or a mixture of different organisms, and considerations for your label. Next slide. So when I think about label considerations, an easy way to do that is looking at an example. This is a made-up label, but one that I have seen on some off-the-shelf products. And there's a few pieces that you should consider as part of being compliant with AAFCO. So one is descending order of organisms by relevant percentage or abundance. So in this case, you see 3.3 billion is greater than the 660 million CFUs down at the bottom. So this would be correct in some regard, but you also see that 3.3 billion is replicated across multiple organisms. So that's one piece that really wouldn't be compliant. The other piece is listing CFUs per serving size. So really this should say CFU per gram. The assumption here is that each of these organisms is CFU per scoop, 1 scoop, or CFU per 2 grams, but that's not really accurately reflected in this. The other piece in AAFCO states that your organisms should be listed not just in descending order, but by both genus and species. So in this case, this is accurate. You can see both the genus and the species listed for each one of the organisms. And the final piece that's missing from here is end of shelf life claim. So all of these counts that are listed are intended to be viable. They need to have a viable statement as per AFCO. And moreover, they need to be verified that they still have those viability counts through end of shelf life. Next slide, please. So when I use the word viable, this is a common term, but one that really just means what are the live cells that are still present. There are fermentation products, and there's labeling constraints around other microbial-based products. Are yeast additives, and I won't get into those, but here in the sense of viable count, we're talking about live microbials. So with that, it's important to consider methodology. Eurofins and many other facilities do offer ISO standard methods, but the reality is those ISO methods for the specific organisms were created for the dairy industry and organisms within the dairy industry. So they're not validated or relevant even to the various manufacturing processes that exist within the pet food or feed industry. We have found that there are many nuances within testing probiotics or direct-fed microbials. I'll refer going forward to direct-fed microbials as DFMs. That's a common abbreviation. But really, any DFM manufacturer out there or strain supplier likely has their own internal methods. And so when we say customer-specific methods, what we're meaning is that particular manufacturer has modified a preparation to optimize the count for accuracy. When we're testing for microbial products, we know there's normal fluctuations. So in chemistry tests, you typically like very tight numbers. But in the world of microbiology, we're also often looking at comparing a half a log difference as acceptable from one product, one lot to the next. And anytime you can reduce that variance is going to help you in identifying how much overage you need to put or formulating your product and formulating it for viability through end of shelf life. The other piece as it relates to customer specific methods is understanding that there should be considerations for how the product was made with regards to microencapsulation, or if the product has multiple species in the finished product blend. There are methods that target specific organisms as a blended product, meaning you can list 2 different organisms and still get a total viable count. Similarly, there's methods that can break through a lipolytic microencapsulation of an organism, but still leave that organism intact. And it's a very sensitive procedure. You can imagine if you're using some type of enzyme to break apart the fat, you don't want that enzyme then to break through the cell layer and kill off the cell you're trying to verify is actually present and countable. And then finally, your label considerations. So we talked about AFCO and the descending order as it relates to your count, as it relates to your serving size, but those are pieces you want to take into consideration as you decide how to test. So for example, if you're going to list your microorganisms in parenthetical value, meaning you're going to list something like a lactobacillus species, and then in parentheses list out each individual species, that's acceptable. However, and that's really the right way to do it. But if you're going to label, as we saw in that example, each specific organism with count, you may or may not be able to do that depending on the constraints of the methodology. So understanding what your objectives are as you test and select method for enumeration is also important. Next slide. When it comes to identification, the question is it genus, is it species, is it strain? And the technology you use to get your identification is dependent on your objectives. I've already covered the AFCO piece and understanding that you need to include both genus and species. And on the right-hand side, you can see a sort of dendrogram where you can see there's bacilli coagulans, etc. This is really just how organisms, the nomenclature of organisms are set up. And the simple way to remember is genus is your group of organisms, species is more specific. So if there's any confusion about what level you need to get, you need to be specific, meaning you need to list that specific organism. Strain is done often by typing. It is often under a proprietary nature. So those typing and proprietary have T's in them. That's strain. That's the difference between species and strain. Strain is more specific. If you are a manufacturer either making an additive or selling an additive, you often have put and invested in some type of research to differentiate your strain from another. And that's valuable. So when you're testing for it, you likely would want to test to strain level. Similarly, if you are a pet food or feed manufacturer that is making a label claim because of that added probiotic, because of that strain, then you want to verify that that is in fact the strain that you have in your product and label it as such. The choice of technology is dependent on how deep you need to go in the genetic profile. Moreover, it's important to understand if you have a raw material or a finished product when you're choosing technology. So there are some technologies that can give you species level identification very nicely, very simply, very quickly, but only in a raw material because it's pure. There's no other added DNA present. We talked about, Pierce talked briefly about allergens and the DNA that's present. DNA can interfere with analyses, and so understanding what test and objective is going to be important. So when you're testing finished product, the same is true. Understanding, do you have multiple species, and therefore may need to run a different type of technology to get that species level information that you need for the label in your finished product. Next slide. And then shelf life. So you do need to verify viability through end of shelf life. This is through counting typically and historically, but there are other technologies that you can employ and have great success with and really provide you an overview. So at the bottom, I realize this is a difficult picture to read the names, but if you look at just the colors, each one of those colors in the bar represent a different organism. Each one of those columns represents a different time point, and you can see a shift in organisms based on the different samples. This type of technology could be used throughout shelf life in a situation where you're including multiple organisms, where you may have both a lactobacillus and a bifidobacterium and potentially other contaminants. And you want to see what's happening in that product over time. It does give you relative abundance. You couldn't use it for labeling, but it really gives you a full picture of what's going on in your product over time. The other piece that's important when you're looking at your shelf life is traditionally, many people are using counts like aerobic plate count or yeast and mold to verify that the product isn't having substantial change or understanding where that product starts to degrade microbially. However, if you're including a Saccharomyces as your positive organism, as your direct fed microbial, a Saccharomyces is a yeast. And so if you have your yeast and mold as your contaminant measure, and that's your only contaminant measure, it may appear that your product is degrading much earlier when in fact that's your positive organism. So when you set up shelf lives with probiotic products, it's important to consider not just the testing for the target organism to verify viability, but also understanding how are you going to test for those contaminants that also impact shelf life and stability. The other piece with shelf life is the consideration for accelerated versus real time. The question is always posed, and I'm sure Carrie and others can attest to the fact that many people want to perform accelerated versus real time, and there's certainly a place for it. But there's really not a place for accelerated when it comes to testing viability and live microorganisms. That being said, real time is always needed, and moreover, there's value in performing a high temperature study, which is essentially equivalent to an accelerated study. We know that probiotics are often susceptible to high temperature, and high temperature can be something like traveling in a large tractor trailer across the country or storing it in a warehouse. So understanding the impact of your stability at higher temperatures as it relates to probiotics is also very valuable. Next slide, please. And the final piece is understanding quality checks. So when you are setting up your probiotic analyses, we've talked about viability and identification and end of shelf life, but you also want to consider methodology as it relates to pathogen testing. So most companies out there, as part of FSMA compliance or other compliance that Gary outlined, are including some type of pathogen review for, say, salmonella. But most of those methods that are executed today are not modified or even validated for products with probiotics. And so if you're submitting a sample that has probiotics, it is important that you let the laboratory know who's performing that that product contains probiotics, and that lab is also making appropriate modifications. If you think about probiotics being present in very high levels, which is often the selling point today, is high levels of probiotic organisms, and you're trying to test for the detection of something like one cell of salmonella, which you would hope if you've got contamination is at low levels, those probiotics could potentially outcompete and the detection of something like salmonella. And what you don't want is a false negative. Certainly we know false positives are bad, but false negatives are equally problematic and should be of concern when you're testing for probiotics or products with probiotics or direct-fed microbials. I already discussed contaminating germs along the lines of stability studies, but again, just emphasizing that you do want to consider how you're testing for other contaminants in a product that contains microbial-based organisms. You want to ensure that the methods you're selecting for contaminants are excluding those intentional added organisms so that you're getting an accurate count and not incorrectly making an out-of-spec or having to claim an out-of-spec result when in fact that's just your intentionally added organisms. The other piece I'll mention here is prebiotics and enzymes. Both of these methods, just like probiotics, are extremely nuanced, and they do have their own challenges. But many people are looking to market their product as having both a symbiotic effect, say a prebiotic and a probiotic, or an enzyme and a probiotic. And so they're formulating these with the idea that if I add a little bit of this ingredient, a little bit of that ingredient, I'll have this whiz-bang, awesome new product on the market, which is great from a marketing standpoint. But the question is, can you, in fact, validate that those ingredients do have a symbiotic effect and are present and not negatively impacting one another in the end or through your shelf life? So as part of formulation and as part of your quality checks, do consider the other added ingredients that may come into play or that you may be considering adding as part of your formulation. Next slide. Okay, I think we had one last slide, but I'll just put a few conclusions out there. So you do want to consider that there is not one method that fits all with probiotic testing. Furthermore, you do want to understand that what your objectives are. And finally, you do want to make sure that you're working with your probiotic supplier to test the right methods and right technology for enumeration, identification, and quality checks. Thank you, Sarah.
Thank you, Megan. Sorry about that. I think your summary slide must have slipped out. We've got over 130 slides in here, so some of them might have gotten lost in the shuffle. But we'll distribute everyone's contact information after the fact, so if any attendees have specific questions for our presenters today, we'll make sure that you have the opportunity to ask those. So as you can see on the screen right now, we're going to get into another question and answer session. Go ahead and enter those questions into the question panel. We've already had a few come in, but I want to make sure everyone has an opportunity. We covered a lot in this session. And then we'll take another 5-minute break before we get into session 3. But first, someone submitted a question early on in session 2 asking if there's any specific reference materials that our presenters could suggest for learning about ingredient risks. Any resources that you guys would recommend? For ingredient risk.
I don't think there are any specific information about ingredient risk. I think the FDA expects all of us to use good common sense and a little investigative activity. Ray, do you have any suggestions?
In terms of ingredient risk, that's a very broad term in what we're trying to analyze for. There are a couple of, I guess, references and guidance documents that FDA does provide in terms of information on pathogenic or like Salmonella or E. coli minimum amounts that should be there, or heavy metal content. That might be something that would be something that would be available. I can't think of a specific one right off the top of my head. Maybe John might have some ideas on those types of risks.
Well, quantitatively assessing risk has been a path that's hard to navigate. To be frank, there is not a lot of clarity specific to pet food in terms of identifying, which risks are important, what levels to regulate, and so forth. Some of our partners are using other country regs. They're using WHO limits for things like dioxins, and they're using non-official guidance documents published by institutes and so forth to get some idea of what constitutes real risk. In general, what constitutes risk for humans would normally apply to pets, and levels that are being set for humans can be thought of as possibly equivalent risk levels for pets, although, of course, the body weight is much smaller in pets, so the risk is obviously much higher for the same level that's found in foods for human. So currently the landscape is hard to navigate, but while I'm not certainly a regulatory expert on risk levels, we do try to help our partners to obtain the necessary resources to at least make some decisions in developing a program for quantifying those risks in their ingredients.
All right, thank you guys. Someone else submitted a question before our presentation that's fit best for our session 2 presenters. They kind of asked if someone could break down the general main risk variations between sterilized raw products and conventional diets. I know there are many. Anybody?
I can try to take a hit at it. It's not my expertise, but the sterilized raw versus conventional diet. Conventional diets have been formulated to contain certain vitamins, etc. And people tend to forget that in a sterilization product of raw diets, you can have some nutrient destruction as part of that process. Sterilized raw diets are good, and because you avoid the risk for contaminants, for microbial problems. But in most conventional diets, especially dry pet foods or wet pet foods that have been heated, we don't see a lot of products. The biggest issue with the bacterial infections normally come in the treats that are fairly openly accessible.